Yes, I really appreciate you coming to spend some of your afternoons with me to hear about our work examining the interactions of climate change and microorganisms in Arctic peatlands. Our work rests on the fundamental question of how the environment shapes communities and is shaped by them in turn, and how we can funnel this understanding into better predictions about ecosystem behaviors. As a molecular microbiologist, my framework for this integration begins at gene expression within cells and extends through populations, communities, and the ecosystem via a web of metabolic connectivity as well as genetic connectivity. Collectively, these connections embody the genes to ecosystems scaling that has been the goal of our field for several decades, and that drive system biogeochemical outputs. Of course, these interactions are occurring within ecosystems that are undergoing global change, which can alter system outputs, sometimes creating feedbacks to climate change. And this highlights what's sometimes called the big biology problem in climate science, which is that even if we could predict the rate and magnitude of humanity's fossil fuel burning over coming years, predicting the full biosphere response is challenging due to ecosystems' myriad interactions. Data from diverse systems show that responses among ecosystem members to the components of climate change, things like shifting temperature, rainfall, nutrients, etc., are not additive, they're emergent. And of course, ecosystems feed back to genes. This is occurring what we refer to as the 3 A's. First, acclimation via changing gene expression, assembly via changing community interactions, and adaptation via changing gene frequencies. Collectively, these ecosystem to genes feedbacks close this loop and create a conceptual framework for characterizing biological integration and emergent ecosystem response to change, and advance predictions for how system outputs may change over time. We proposed to the National Science Foundation's new Biology Integration Institutes program to apply this genes to ecosystems to genes framework at a deeply studied Arctic ecosystem. And last year we were selected to be just one of 4 in the inaugural group of 5-year, $12.5 million institutes funded by NSF, with ours led here at Ohio State University. To accomplish this work, eMERGE integrates a number of biological disciplines along with modeling, computational science, remote sensing, education, and the social sciences through a collaboration of team science of more than 90 scientists from 15 institutions from Australia through diverse organizations in the United States and all the way to Sweden. In addition, we integrate across the 6 components of our institute with this really important underpinning or overpinning, as it were, of team science training through data management all the way down to outreach. And a shout out to the Center of Microbiome Science here at OSU for partnering with us on education and training through their webinar series that was very well attended by hundreds of scientists from around the world this spring, and through which eMERGE was able to share some of our toolkit of tools as well. At its heart, these ideas, collaborations, and training programs and the data that we have actually grow out of a decade of multidisciplinary research spanning 4 main projects focused on methane cycling at Stordal and Meyer and funded by the Department of Energy as well as NASA and the National Science Foundation. At its heart, eMERGE addresses twin grand challenges in biology. The first is one of NSF's big questions, which is defining the rules of life that produce phenotype from the interactions of genotype and environment. The second is the largest challenge our species has faced, managing a rapidly changing biosphere. And the connection between these two is precisely the challenge of biological integration. unifying mechanisms and feedbacks across scales into improved predictive understanding. So it is within that context that the overarching scientific goal of eMERGE is to identify emergent principles of ecosystem response to change by applying this swoop of genes to ecosystems to genes framework at a model consequential ecosystem. What is the ecosystem? You can see it in the background here. This is Stordalen It sits above the Arctic Circle in northern Sweden. That means that it sits at the southern edge of the discontinuous permafrost zone, which means that climate change is happening very quickly there. Notably, for this site, the research station that's there has been in operation for over a century, which is totally unique for an Arctic research site. And that means that there's a lot of on the ground measurements that stretch back since long time before I was born, and probably before everybody in the audience was born. So that's, oops-a-daisy. That's very exciting and powerful for establishing the localized context for our measurements. And the Stordalen, which is adjacent to the station, has been intensively studied since the 1970s. It was actually the site of some of the very first field methane measurements and a focal site for the International Polar Year surveys, etc. So it's a great site to be working at. And part of this long-term effort there means that it has extensive in-place instrumentation, which you can see here through these eddy covariance towers. You might have heard of these, also called eddy flux towers, automated gas sampling chamber systems, lots of machines that go bing for measuring the carbon and other greenhouse gases that are emitted from these systems. So it's a very powerful framework for us to be layering on top of. And we started doing this work there over a decade ago. And because it sits where it does at the southern edge of the discontinuous permafrost zone, the thaw that is happening there is on the scale of individual PhDs, as you can see here where the boardwalk has collapsed. An important thing to understand about permafrost, which you might have seen in the news when you see reports about infrastructure in northern sites kind of failing and collapsing, is that the permafrost, as it thaws, actually settles and collapses away. Sometimes you can have big pieces of it kind of cleave off, and sometimes it just sort of compresses down. And that means that, just as you can see with this boardwalk here, now slumping, that same thing happens to pipelines, to roadways, to entire cities. So this is part of the global trend of permafrost loss on a warming planet, with large projected decreases by the century's end. And this range of lines reflects a range of different scenarios for humanity's continued burning of fossil fuels, with the uncertainty around each line reflecting the uncertainty about the biosphere response. Now, this is problematic that these lands are thawing, because a third to a half of the planet's soil organic carbon is locked up and stored in these northern soils out of biological action. But now, and this carbon has been frozen for hundreds to thousands of years, but now the freezer is failing. And as that's happening, we see increasing emissions of the very potent greenhouse gas methane from these systems, from some of these systems, not all of them. Methane is about 30 times as potent of a greenhouse gas as CO2. And so at the site we're studying, the transition from intact permafrost to fully thawed peatland is associated with about a 7-fold increase in global warming potential on a per meter squared basis. So it's a big impact in terms of that positive feedback to ongoing change, okay? So this is a consequential ecosystem. And we have been looking with what we call an ecosystems biology characterization of carbon cycling along this thaw gradient. And we're really interested in carbon cycling because of that fact that you heard just a minute ago, which is that these kinds of systems store so very much of the planet's carbon, and that carbon is now being released into the atmosphere, where it can then accelerate climate change. So we've been looking at the site for carbon gas fluxes and isotopes. Fluxes just mean the amount coming off the top. We've been profiling the vegetation, been looking at the microbiomes there, and looking at the geochemistry over this time period. And we really consider this a sentinel ecosystem of climate change because of where it is poised. And so we are hoping to learn as much as we can from it as it is changing, to be able to cross-inform our interpretation of what's going on elsewhere in the biosphere now and in the coming decades. So our ecosystems biology approach to looking at all of the things in these systems has given rise to data streams that integrate across space and time, and specifically span a range of kinds of data, and allow us to pair targeted field sampling, because you saw the state of that field site, right? Every year we go out there, we feel terrible about taking any cores from it because the place is collapsing, right? So we try to tread very gently on that ecosystem and take the minimum number of samples that we can and pair that with a range of non-destructive measurements, as well as ex situ incubations, things like culturing, incubations, and bioreactors. And then these are integrated through a series of models, producing site and Meyer-wide portraits of methane cycling. So this generates really powerful site-specific reference libraries of the chemicals, genes, and organisms, and those empower our interpretation. So in order to understand ecosystems where our measurement capabilities are now pushing the cutting edge, so that we're able to see types of molecules and types of organisms that have never been seen before regularly. To be able to interpret what those mean and how those fit into an ecosystem context, it really helps to have a lot of carefully assembled reference data for that site, where we've applied a lot of different tools to do the best job we can in characterizing, say, the molecular formula and most likely structures for the chemical compounds in the dissolved organic matter, for example. And so by studying this single site so intensively for this time period, we've now generated those kinds of reference libraries that really represent these amazingly powerful shoulders of giants to stand on in terms of our data to be able to do a lot now. So what have we found? Well, by sequencing about 1,000 different samples from metagenomics now, we found a lot of new microbes. And those have been recovered as genomes for more than 10,000 different assembled microbial genomes. Metagenomes are where you sequence all the DNA from a community that you can extract. And so that will include some plant community material, some little microinvertebrate stuff, maybe some things from some reindeer poop because reindeer come through there, all of those different types of things. In this study, for the first decade, we focused on the DNA level at the microorganisms, which we're defining as archaea and bacteria. Okay? And so this represents an evolutionary tree of all of the lineages that we have found at this site, and each of the different branches of the tree represents a different phylum. And the red portion is novel organisms, and the gray portion of each of these little pie charts is things that were previously known. And so you can see that there are entire phyla here that never had genomic representation before until this study. So that's really powerful just in terms of thinking about biodiversity. And you might be from industry, you might be here from industry, and you might be thinking, oh, I wonder if there's a novel cellulase that might be useful for biofuels in there. That's a great question. You might be from medicine. You might be thinking, I wonder if there's any new kinds of antimicrobial compounds in these different genomes, because these organisms living out there in this intense environment might have new ways of engaging in warfare with one another. So they might have new types of antimicrobials. Also a great question. This represents this treasure trove of data to be mined for all kinds of different questions and is publicly available. The questions that we are interested in and that we have the capability to ask with the team that we have funded are really focused on those climate feedbacks. So in addition to it just being a whole heap of genomes of these different microorganisms, which we can characterize based on their evolutionary identity and their genetic gene content that can hint to us what their metabolisms are most likely to be. So that's useful in and of itself. But what's super cool when we think about that scaling up to ecosystem outputs is that these, what we've recovered now, the latest number is this 10,000 number. This is going up rapidly all the time. It's very exciting to watch this number go up. This represents 75% of the cells on the mire at the genus level, which means we're doing an increasingly good job of being able to holistically represent the different kinds of lineages that are there at the genome level. Now, why is that important? It's important because just knowing which organism is there does not tell you what it does. So you can have two very closely related microorganisms that do different things, quite different things. And so you really want to be able to have that genome or a genome of something very closely related that you can start to use to interpret the role of that organism in the ecosystem, okay? So having those genomes recovered is a big deal for understanding the overall microbial community and indeed ecosystem function. So this is very exciting to us. We've got viruses. Notably, these are viruses that infect microbes. We're not talking about viruses that infect humans here. We're talking about viruses that infect microbes. Again, thousands upon thousands of them have been recovered. The genomes form them, not the actual viral particles themselves, but the genomes for them from the DNA metagenomes that we've sequenced. And we see that these viruses infect different types of microbes and carry different types of genes. There was a question that one of you excellent audience members asked ahead of time that got passed on to me, thanks to the great team there. And it was, what about viruses that make humans sick? And some of you might have heard about those kinds of viruses from permafrost associated systems. Where we are most worried about that is in places like graveyards. You might have heard stories about graveyards in Alaska, where bodies were buried in the permafrost. The permafrost is now thawing, and old diseases that were present in those bodies are now being kind of released to those ecosystems. That is a specific thing where you have the most likelihood of having those kinds of diseases still kind of archived. In places like our mire wetland, that's not a thing that we see. Fortunately, there's not bodies there. So we don't see pathogens in our data. Okay, so in this lovely treasure trove, and I'm just going to give you a few examples here. In the interest of time, I want there to be time for discussion, and I didn't want to put up a ton of data because there's so many different stories to tell, and I'm not sure what all of you will be interested in. So I'm just going to give you a few examples of things. So in this treasure trove, we found a new methane producer, a brand new type of methane producer microorganism that hadn't been seen before. We were able to recover its genome. It is the dominant methanogen. Methanogen just means methane producer after thaw happens. So this red slice of this pie chart here represents that novel methanogen. It's highly active. We can look at the RNA and protein from this site to see that not only is it hanging out and it's there, but its genes are actively expressed, including its genes for methane production. And the postdoc who was leading this work was able to go into the sequence databases that are present out on the web and at different repositories and see that the signature of this organism, this novel methanogen, had been seen all over the place before, but nobody knew what it was. And this is a really common thing in microbiome studies, that different lineages are seen that we don't know what they do. And so people just report the sequence, put it into the repository, and don't tell their story about that thing because they have so many other things to look at in their data, and they're asking very specific questions, just like in our case, when you think about, novel solutions to biofuel production or novel solutions to antimicrobials. Those could be in our data, but that's not what we're looking at. Similarly, there was this novel methanogen in lots of other data sets all over the world from similar kinds of sites that were either measurably methane producing or likely methane producing, and it just wasn't measured. Okay. So this new type of methane producer is active at our site, responding significantly to climate change, and globally ubiquitous. Okay, that's impressive in these kinds of sites. In addition, it's predictive of the isotopic signature of the methane. When methane comes out of the ground, it has like a signature based on the isotopic characterization of the carbon and the hydrogen. And it turns out that this one lineage is really tightly correlated to that at our site. And that is relevant to distinguishing human-made methane from natural methane in the atmosphere. So one of the ways that people try to figure out how much fossil fuel burning has led to methane is by going up and sampling in the atmosphere and looking at the isotopic signature of methane in different places and globally. And then relating that to reference numbers that scientists have made for, like, what does an average methane look, what does an average wetland source look like, what does an average fossil fuel burning source look like, what does an average cow toot look like, and they try to put all of this together and say, well, can we back out what the contribution of these different end members must be based on the source isotopic signatures and what we actually observe? It turns out that when permafrost thaws and makes wetlands, like at our site, the signature of those wetlands is not the same as like the standard wetland signature. So that means that if you apply the standard wetland signature as your assumption when you're looking at atmospheric levels, you end up underestimating, and sites like ours are important, you end up underestimating the industrial contribution, the human contribution to methane sources. So there's kind of two pieces to this: correcting those reference numbers to reflect the increasing role of wetlands like ours, and recognizing that that seems to be driven by particular microorganisms like this novel methanogen that seem to be driving a different isotopic signature than sort of our typical temperate area wetland. So this new methane producer also, and again, this is just one particular kind of vignette here, this new methane producer also has an ecologically and evolutionary conserved partner that the team discovered that seems to be able to help it make methane as the permafrost thawed. So as you have permafrost thaw, you see different lineages of both the methane producer and its partner showing up in a consistent way in different stages of permafrost thaw. And they have kind of ecologically matched here, they're ecologically matched here. And when you do the metabolic reconstructions based on their genome of like, what do we think these organisms are doing, you can see that there appears to be a partnership. And so members of the team are now working hard to try to culture these organisms, to grow them in the lab, to see, can we observe this actual partnership? In addition, they're using cool molecular methods to take samples from the site and see if they can trace this out microscopically, even if they can't grow them in the lab. Because as you may have heard, the majority of microorganisms out in the environment are resistant to being grown in the zoos of our laboratories. In addition, there's community-wide changes in carbon processing after thaw. There's community-wide changes in the optimal growth temperature of the microorganisms after thaw. You can see a genomic signature of an increase in the optimal growth temperature of those microorganisms before and after thaw at the ecosystem level, which is kind of remarkable. So we're in the process now of testing that experimentally through incubations outside the site and seeing if our genomically predicted optimal growth temperatures are borne out. There's been a lot of other research in other sites and other isolates, other microorganisms that kind of have validated that relationship, but now we're trying to see, like, is it valid at our site? Because this is a cool finding. There's also really high temporal variability in methane emissions, and that's what's shown here. So on the y-axis here, you have the methane flux, and on the x-axis, you have the time through the growing season from May to around now, to November. And you have unthawed sites in brown, where the permafrost is still intact, partially thawed sites in green, and fully thawed sites in blue. And each of these different lines represents a different year. And in this case, it's just 6 years. But what you can see is that there's a great deal of variability, not only from year to year, but from sort of month to month. And that's driven by things like temperature, atmospheric pressure, plant growth, et cetera, rainfall. And this really highlights that things like those fancy measurement systems at this site that are measuring things continuously make a big difference. Because if you just go out and take a point measurement here and then a point measurement over here, it's going to be very difficult to then extrapolate accurately to the overall growing season. In addition, we found that there's new chemistry occurring. So reactions, this is just one particular example that is kind of intriguing and has come out just in the last couple of years, led by the chemists on the team, that reactions known from the leather tanning industry are actually occurring at apparently large rates, large amounts at our site. And it looks like in other sites like our site, other kinds of peatlands like our site, and that's consequential. And so these particular reactions are not microbially mediated. These are happening abiotically. And even though they're known from this industrial process at high heat, it looks like they're occurring under natural conditions. And as they're occurring, they're actually locking away nitrogen, which has impacts on, of course, the plants and microbial communities there. And that those reactions are changing as the plant community changes during permafrost thaw. We're learning more accurate ways to model these ecosystems as we're trying to funnel down all of these diverse observations into more accurate models. We're also learning that we can use satellites to reasonably infer microbial communities. And so this is work that was originally funded in a grant to our co-director, Ruth Varner from NASA, to use satellites to look across the Arctic at different types of communities and see if we could link that to some of these greenhouse gas emissions. So here's just 2 quick slides on that. So here's an image of the WorldView 2 satellite, which is the main satellite being used, whose data is being used in this work. And really, when we think across the Arctic, like this huge expanse here, there's very few boots on the ground in most of those places. And so if we're trying to infer what the methane emissions is now and in a couple of years from these sites, that is a tough challenge, but it's really consequential because knowing the trajectory of our overall climate response depends a lot on these kinds of ecosystems. There is an appreciable feedback here, potentially. Okay? So what Ruth asked was, can we predict system outputs and feedbacks via remote sensing? And so this is the WorldView 2 image of our Stordal and Meyer. You can see the boardwalk system through there. If you look closely, there's a shack. Here's our chamber system, and there's another shack right there that houses all those fancy pieces of equipment. There's eddy flex towers all across this landscape. The brown areas are the places where the permafrost is still intact, and you start getting greener areas when it starts to thaw, and then increasingly green and boggy areas when it's fully thawed. So what Ruth's team was able to do, and this work is now being built upon and continued through Emerge, is translate that worldview to satellite imagery, which is just, this is the sort of visible spectrum, right? So you can see this here. And they can translate that algorithmically into different land cover types. And this is actually the algorithm that they developed iteratively to do this is very accurate at the landscape scale. So if you go out and you point check and see how good of a job it did, it's like 98.5% accurate in parsing that satellite data into land cover type. Okay, so does it look like intact permafrost? Does it look like partially thawed? Does it look like fully thawed, et cetera? And then you can do two different things. If you just want to understand what the emissions might be across a big swath of land, you might just be able to do what's called painting by numbers. So we know a bog in this kind of part of the Arctic tends to emit this amount of methane. And we know that a fen, which is a more thawed stage of these kinds of particular ecosystems, emits another amount of methane. And so we can just apply those averages to everywhere that our algorithm tells us is one type of land cover versus another. And so that would be sort of this paint by number approach. And you can do that, and we're doing that. But, and other people are too, but given the ongoing nature of climate change, we need mechanistic models that don't just paint by number using existing averages from these kinds of sites, but try to kind of get under the hood and include how the system response is changing, how the plant communities are changing, and tries to layer in some of these intricate feedbacks of genes to ecosystems back to genes that we're trying to describe. The first step of that is trying to accurately use things like the satellite data and our on-the-ground measurements to drive models that represent some of that complexity rather than just a paint by number approach. And so this is doable, and we're making really rapid progress on this, and it's very exciting. So in summary, I've introduced you to this idea of the genes to ecosystems to genes foundational structure for looking at these kinds of ecosystems. I've introduced you to our, what we call the model ecosystem. You've heard about model organisms, things like Drosophila and yeast. Drosophila is fruit flies, and yeast and mice, organisms that have been transformational in the last 50 years of biology in helping us understand the rules by which cells work and organisms develop. I believe that there is a movement afoot now in ecology to try to do the same thing as model organisms have done for kind of cellular and developmental biology, to do that for ecosystem biology using model ecosystems. And these are ecosystems that are very well studied and ideally are consequential to things we care about. And so at our site, we're applying this framework to our actively thawing consequential site to generate a whole boatload of beautiful and powerful data. And that is allowing us to tell a great number of stories about changing carbon cycling. And I introduced you to just a couple of those. I introduced you to this novel Methanogen, whose name, if you're interested, is Candidatus methanoflorans strodallenmeirensis. And so methanoflorans means methane flower, and strodallenmeirensis is after Strodallenmeyer, where it was discovered. And I introduced you to the fact that, in fact, even though it was discovered at Strodallenmeyer, it's present across a range of different sites, and not just Arctic ones, but temperate and tropical ones as well. And we talked briefly about the fact that it seems to have a buddy that it always co-occurs with that helps it survive and flourish in these environments. We also just talked briefly about the fact that it seems like we are able to, and I didn't take the time to kind of get into the weeds and show you this data, be happy to do so. But it seems like we can reliably, consistently measure the plant communities above ground using the satellites, binned into different types of kind of characteristic plant communities that represent different thaw stages. And those reliably predict the below ground microbial communities. There's error in that, but overall, it's a pretty strong relationship at the level of our entire mire where we've gone out and taken different samples. And so that's super cool that we see that consistency of the above ground plant communities and the below ground microbial communities. And that starts to allow us to have some confidence in upscaling, because the organisms that are doing these processes of making these greenhouse gases seem to be kind of reliably linked to the above ground communities. And lastly, I introduced you to our new Biology Integration Institute that just launched last year, just over a year ago. And despite the challenges of the pandemic times, is really gaining traction and doing some very exciting things. And now we've got about 4 more years of funding to really see where this takes us. So I'd like to just flag that right now, of course, as most of you know, it's the 26th UN Climate Change Conference of the Parties, COP. That's going on, and there's world leaders that are working towards mitigation and adaptation scenarios and solutions. And in our little institute, we care a lot about this. You know, we're seeing it happening all the time in our data and in our field site. And what we are endeavoring to do is help provide a small piece of the solution in helping provide predictive mechanistic understanding of ecosystem response. Because as a species, we can't manage these natural ecosystems if we don't know the rules that they're playing by. And the rules that they're playing by in a static moment is not how things will go as conditions continue to change, because of these feedbacks, the 3 A's of these feedbacks. So with that, I'd like to thank you so much for your time and acknowledge the diverse partners that Emerge has and our funding sources. And I'd be delighted to take any of your questions.
Thank you so much, Virginia. Yes, to everyone in the audience, please feel free to submit your questions. You can use that Q&A feature at the bottom of your screen, and we'll be happy to answer any of those. Virginia, sort of one question off the top. I was curious, with the permafrost thawing so quickly, kind of how is the infrastructure at the research site and kind of the work that you all are doing, how has that changed since the ground is thawing beneath your feet?
Yeah, that's a great question, Clara. Thank you. So this is actually something that we're having meetings about on sort of a weekly and monthly basis right now, because one of the things that has started to have to happen is actually putting floats under some of the infrastructure. So for example, we probably are going to need to move the auto chamber system, because instead of spanning intact permafrost through partially thawed permafrost through fully thawed permafrost, now almost the entire chamber system is in thawed areas. So if we want to still capture the gradient, then we need to move the whole thing. But things are changing so quickly that, and the other thing is the actual shack that's housing all the instrumentation is sort of falling into the bog. And a lot of the things, like you'll leave little flags behind to be like, we sampled here last time, let's not sample the same place. And you come back and the flag has been eaten by the mire. And so one of the solutions is actually putting like pontoon kind of floats underneath some of our infrastructure, so that as the system changes, our structures can be supported. And we try really hard, you know, that one of the senior researchers on the project has worked at this site for a really long time, and we sort of affectionately call him the kind of guardian of the mire, because he's like, if you're ever out there with him, he's like, stay on the boardwalk, don't stray from the boardwalk, because every time you step a toe off the boardwalk, right, it impacts the system. The boardwalk itself impacts the system. All of our measurements, if they're invasive at all, are impacting it.
Wow. A question here from Mark: is Antarctica undergoing similar change?
That's a great question. Yes, the poles are changing. There was a figure earlier on in my talk. I won't bother to share it right now. It's easily Googleable, but if you look at the climate change, the temperature change globally, and the predicted temperature change globally, you will see that it's happening disproportionately quickly at the poles for a variety of reasons. And so there are these two, that's always red for the warming, and you can see that there's strong red blobs at both of the poles, and then also red everywhere else, but it's really happening at the poles. So yes.
That's great. And then when you're talking about thawing permafrost from sort of the completely unthawed to completely thawed, what's that timeline look like at the research site?
Yeah, so it's variable. And that's because it depends on things like water flow through the system. It also depends on sort of the plant community. How much insulation is the current plant community providing? If the water is flowing through that particular area, it really speeds up the thaw. So you can have a transition from fully intact pulsa to like fully thawed fen within 5 years for sure. However, there's other places where the pulsa is staying intact, the permafrost part is staying intact and isn't thawing yet. So that's one of the powerful things about being at this discontinuous southern edge of the permafrost zone, because you can see that kind of heterogeneity.
Wow. And a question here from James, are you seeing any vegetation changes in the mires, and are there any changes in the interactions between the microbiota and the plants?
Yeah, and this is actually one of the most fascinating things. And so we've had sort of arguments and friendly arguments and bets over the years about like, are the microbes, slaves to the plants, or like, what's the driver? What's the ultimate driver of all of this? And so as the permafrost thaws, the hydrology changes, as you saw. The water table gets closer to the surface. Okay. And sometimes even goes above the surface, and then you get these thaw ponds. The plant communities change along with that. So you go from having these cute little herbaceous shrubs, things like cloudberries, which are a delectable thing that grow locally there, and they make jams out of, and very tasty. So you go from having little ericaceous shrubs like cloudberries to something that's really dominated by sphagnum mosses. And sphagnum mosses have very interesting chemistry. And many of you will probably have seen bog bodies. You might have seen that in the media at different times. So there's like, if you Google Tolland man, you can see pictures of preserved human bodies in different peat bogs in Northern Europe. And they're very well preserved. It's almost like mummification, or mammoths preserved in glaciers, that kind of thing. They're very well preserved because the sphagnum moss creates a more acidic environment, and it also has chemical preservatives in it that can prevent the rotting of material. So you can find, again, we don't see this at our site, and it happens on sort of like an archaeologically random basis where people, had a mishap hundreds or thousands of years ago and got buried in a bog, but periodically people will find in peat bogs different interesting kinds of bodies. And so I mention that to say that the preservative compounds in sphagnum are impressive, and there's a lot of those coming out into the site. And then as you thaw further, you get sedges, which look kind of like grasses. And those organisms are pumping, or those plants are pumping all kinds of delicious types of carbon down into the subsurface. And so they're leaking it out through their roots, and it's almost like they're farming their own microbiota underground. They also have roots that are almost like straws and aerate the ground underneath. So the plant communities are absolutely controlling the physicochemical space below ground in a way that intricately interacts with the microbiota.
That's great. Again, if anyone has any questions, please feel free to drop them in the Q&A. But Virginia, I was wondering, you mentioned, I think you have 4 more years on this specific project. Sort of what are you most excited about and most looking forward to in the next stage of the project?
Oh, that's a great question. I would say there's a couple of different things. The microbes from space part is just, How is that not just like so exciting? When Ruth first came to me and she's like, do you want to write a project, a proposal to NASA together? Like, oh, yes, please. So that's really fun. I would say also co-director Ruth Varner, who's at University of New Hampshire, she just received an independent grant from the National Science Foundation to run a graduate training program that actually partners with Indigenous communities throughout the Arctic, but also heavily connected to the Sami people of Northern Scandinavia, who are the traditional reindeer herders. And I am very excited. She's spent, for that work to start, she's spent years developing relationships with local Sami, and she's in the field a lot more. I don't get a chance to go to the field as much. And so she's now built on those relationships and partnered with folks there to find out the kinds of things they're interested in and the kinds of education they would like for their own communities to have in partnership, and also the kinds of education they feel like they can offer and what their kind of research and educational priorities are. And so she has developed this beautiful graduate trainingship around that, and National Science Foundation just funded it, and that's just starting this year. And it overlaps in a very powerful way with what we're doing in Emerge on the research side. We have an RU program for undergraduates, and then she's got this graduate trainingship that partners with the Indigenous communities there. And so collectively, I feel like this is a sort of powerhouse combination of excellent cutting edge research, really strong education and training in a context that acknowledges the communities that have had centuries long stewardship of the places we're working. So I think that that's overdue, and I'm really glad that's starting.
That's wonderful to hear. All right, well, we don't have any more open questions right now. If you have a burning question, please submit that here. But Virginia, thank you so much for your time. If you have any closing thoughts you wanted to share with the audience before we close out, feel free to share.
Thank you. I guess I would say, so if there's any undergraduates in the audience, we are running an REU program next year. We also have an early career researcher program, which is actually open to any researcher who's interested in this kind of stuff. You don't need to be early career at all. And that's going to happen next summer. If you are a data scientist of any kind, and you want to know about cellulases or antibiotic resistance genes or anything, please come play in our data. We have this beautiful data and far more questions to ask than we have funded people to ask them. And we're just looking at a little piece of all this. So please, we would love to play. Come see us.
Wonderful. Well, thank you so much, Virginia, for sharing your time and expertise with all of us. And thank you to everyone who was able to join us today. We'll see you soon. Stay safe and go Bucks.